So I'm Konstantina, in Greek Chachlaki, for the rest of the Europeans usually is Chachlaki. Thank you so much for having me here today. Thank you, Sandra, thank you, Marita, and all the GRIN family for this kind invitation. I must say I'm a bit of an imposter because I'm not really working on GRIN disorders, but I'm working with a molecule that I hope that you will find interesting, which is nitric oxide. And just a very few words for me, since no one knows me, but you all seem to know each other. So I'm Greek. I have a background of molecular biology, and I started working on neuroscience during my master's degree, and this is where I came across this molecule, nitric oxide, that, as you can maybe see in the continuum of my career, it became a bit of an obsession. So then I did my PhD on moving from the neuropharmacology, let's say, of the molecule more to its neurophysiological actions. And I also passed through the genetics that could involve nitric oxide deficiency in humans. And right now I have a little group that inside a big team, as you will see in the end, in the University of Lille. And I have the honors to coordinate a clinical study around the use of this molecule for Minipuberty that I'm going to talk about. So what is nitric oxide? Actually, it's a very interesting molecule because it can be actually produced by 3 different synthases. There is the neuronal form of the nitric oxide synthase, the endothelial one, and one that is inducible. Today we are going to focus more on the neuronal form of the synthase. And this molecule is actually very cool because it can be produced by the 3, and then it can freely cross any type of membrane. And once in the interior of the cell, it will act either by promoting the production of a downstream messenger, cyclic GMP, which is a molecule with broad actions, or by performing some type of post-translational modification, that is DS nitrosylation, for which I'm not going to talk at all about today, but it's a very interesting mode of action because it has been linked with the pathophysiological actions of nitric oxide and its involvement in the inflammatory phenotype. So nitric oxide can be actually, and the NOS neurons are actually found almost everywhere in the brain. You can see them here in white, in regions that most of you are interested in, like the hippocampus or the cortex. But today I'm going to mostly focus on these regions, that is the hypothalamus. So it's a tiny structure that is found on the bottom of our brain and has the size of our thumbnail, basically. It's a tiny structure. And more specifically, I'm going to focus on this region here, which is the preoptic area of the hypothalamus. And why is that? Well, first and foremost, because the most interesting population, the biggest population that we found in the hypothalamus was found in this region. I mean, you will see also other images later, but you can see that there are many cells there. And the second and most important, maybe, reason is that in that area we have the main regulators of reproductive functions that are the gonadotropin releasing hormone neurons, the generate cells. So there is a 3-level axis starting from the hypothalamus, moving to the hypophysis and then the gonads, that is going to be regulated. But this tiny population of cells, there are only a couple of 100 neurons in the rodent brain and a couple of 1,000 neurons in the human brain. So it's really a very tiny population of cells. So what they're going to do is that they are going to release the generates peptide that is going to travel in the hypophysis to actually promote the release of the gonadotropins, which is the luteinizing hormone and the follicle stimulating hormones, that then they are going to act on the gonads, so the ovaries and the testes, in order to promote the production of the sex steroids, which are the estrogen, progesterone, testosterone, and the gametogenesis. What is actually interesting to note is that these sex steroids will then act back onto the brain to come and regulate the function of generates neurons. And these generates neurons will not only receive signals from the sex steroids, but also from other type of peripheral hormones, like, for example, leptin, that is secreted by the adipocytes and is a metabolic hormone, and a bunch of other hormones and also neuropeptides that are going to come in order to collaborate with the degenerate neurons and promote not only reproduction, but many other physiological functions, such as metabolism, sleep, thermoregulation, and many other functions that are organized by this bunch of cells. And one of the populations, obviously, that is found all around the degenerate neurons, and we're going to focus today, is the cells that express this neuronal form of the nitric oxide synthase. And you can actually appreciate that in both in humans, but also in mice, these NOS neurons are actually found all around the degenerate cells, and they create this type of anatomical connections with these guys. And what is also actually very interesting is that the NO cells are also communicating with the rest of the signals that are important for the function of degenerates, including, for example, the estrogens, because they abundantly express the estrogen receptor, including kisspeptin, which is another key population controlling reproduction, that is the main actual activator of the degenerate neurons, and leptin, that, as I mentioned, plays a key role in metabolic function. So it means that they can actually incorporate many different signals to come and communicate with the degenerate cells, giving them information about what is happening in the periphery. And what is actually important maybe for this talk today is that these NOS neurons that are found in the preoptic area of the brain are actually glutamatergic, meaning that they themselves, apart from nitric oxide, they can also produce glutamate. And why is it important? It's not only because glutamate is interesting for the GRIN network, but it's also because the production of nitric oxide is itself kind of regulated by glutamate. And what I mean by this is that the activation of the NMDA receptor through the influx of glutamate, through the activation by glutamate, is going to induce basically the influx of calcium that is going to come through the calmodulin kinase to promote the anchoring of the NOS protein through the PSD95 on the NMDA receptor. And when found in this conformation, the neuronal form of the nitric oxide synthase can actually be phosphorylated in this residue with this serine 1412, and this is going to promote the activation of the enzyme and the production of nitric oxide. And as I said, this nitric oxide will be then able to freely diffuse across the membranes, and it's going to bind to the only known receptor so far, the guanylate cyclase, in order to produce cyclic GMP. And then this cyclic GMP is going to be, as a second step, controlled by the presence of phosphodiesterases that are going to degrade and like that stop basically the nitric oxide downstream signaling. And this is something that can be modulated by all these members of the degenerate network that I previously mentioned, meaning that all of these, estrogen, leptin, kisspeptin, and many more, like insulin even, they can promote the phosphorylation of nitric oxide synthase in this residue, meaning that they can control the activation of the enzyme. And here I have an example of estrogen showing basically that the increase of the phosphorylation of the enzyme, so meaning this is the proxy for the activation of the enzyme, actually fluctuate across the female estrous cycle, being maximal at the day of the ovulation, meaning that estrogenic signals will control basically the amount of nitric oxide that is present in this hypothalamic area. And apart from the, if you want, the control by estrogenic levels throughout the estrous cycle, there is another level of control that happens developmentally. And as you can appreciate here you have on the left the female and on the right the male. You can see that the amount of NOS neurons does not change across development. So here we are in the first 2 weeks of life, as you can see, postnatally, so this P7 is from postnatal day 7. But what changes is the activation profile of the enzyme that seems to be activated the 2nd week of life in the female and around the 3rd week of life in the male. And when we went back a bit into the axis to see what is happening basically in the rest of the axis during that period, we realized that this period corresponds also to the first rise of the FSH levels, meaning this activation of the pituitary in the females. And at P23, as you can see here, it corresponds perfectly once more with the FSH levels, meaning that basically degenerates and NOS neurons kind of mature at the same time. And this period is key, and this period is called mini-puberty, and is the period for which I'm going to talk today. And it's a period of life that actually arrives in all altricial species, including rodents, but also humans. In humans, we know that it arrives around the first to second month of life in the little girls and the little boys, and is basically corresponding to this first activation of the hypothalamic pituitary gonadal axis that we see with this release of the FSH and LH hormones. And this period is going to stop around the 6th month of life in baby boys and baby girls, and the axis is going to stay silent throughout the childhood, and then will be reactivated at the time of puberty, and from that point on will continue to be active for as long as we are fertile. We don't know exactly the importance of this axis for later on development, so we went back to our mouse model to try to start manipulating a bit the axis and understand what could be its importance for the developmental processes. So here you can see basically mice that are either wild type or completely deficient for the neuronal form of the nitric oxide synthase, and you can actually see here that the FSH level, so this mini-puberty, is actually exacerbated in the absence of the neuronal form of the nitric oxide synthase. And apart from the fact that we have this exacerbation, we also have a longer duration of the activation of the axis that normally in mice it's supposed to shut down in the 2nd week of life for the females and around P30 for the males. So you can see that in both situations, basically we have an alterations of both the amplitude of mini-puberty, but also the duration of mini-puberty, meaning that nitric oxide is actually important for controlling this strict window of developmental maturation. When we went back to a bit to the humans to see an equivalent, maybe, example of an alteration of mini-puberty, we used basically a cohort of genetic generates deficiency. Why? Because these people with a genetic generates deficiency, so a syndrome called congenital hypogonadotropic hypogonadism, they have absence mini-puberty, delayed or absence puberty, they are infertile, and they have a bunch of different comorbidities that are non-reproductive. And we actually identified for the first time the presence of NOS1 mutations, so 5 different mutations in the NOS1 gene, coinciding with the generates deficiency, but also with nitric oxide deficiency. We were able to measure the nitric oxide in vitro in these different patients using a very actually super sensitive technique. We have a nitric oxide biosensor that we can use to do that, and we actually basically demonstrated for the first time that generates deficiency and NOS deficiency coincide, and they can play, both these neuronal populations can play a key role for reproductive function. And what is of note here is that apart from the reproductive phenotype that CHH patients demonstrate, they also have, as you can appreciate here, many associated phenotypes that are non-reproductive. We have hearing loss, we have intellectual disability, we have obesity in some cases, and this is a characteristic of many other genes that are associated with CHH, but obviously we were very interested to check back what is happening with the NOS deficient mice and this type of comorbidities. We started from the reproductive comorbidities, and we actually show that this alteration of mini-puberty that we had was also associated with a delayed pubertal onset in both females and males, and then in subfertile phenotype, in some extreme cases, complete infertility, with an alteration of the estrous cycle in the case of the females, and an aberrant type of pituitary function in both sexes, with, as you can appreciate here, very, very low plasma LH levels, meaning corresponding to this subfertile phenotype. We started taking the non-reproductive phenotype. We started with some hearing tests. So you can see here basically the ABR thresholds that seem to be quite altered in the Enos knockout, meaning basically that they had a higher threshold of sound intensity in order to evoke an auditory response, but also they had increased latency, meaning that they had a delay in the transmission of the signal to the cochlear nucleus. We also tested the olfaction of these guys, and we saw that they had an aberrant response to social odors, meaning that the female mice were not able to recognize the male odor, and vice versa. While what we saw here that was mentioned earlier in the talks today, we saw a kind of a sticky phenotype, I like this term, meaning that in an dishabituation test, the Enos deficient mice really were not able to perform the dishabituation. So they were really obsessed by the new odor, by the odor that was presented to them, that we concluded eventually that was more related to a cognitive defect rather than an olfactory one. And indeed, we performed this cognitive test. So we had a decreased response to a novel object recognition test, pointing out to a problematic recognition memory. And we performed also a different battery of executive function and cognitive flexibility tests that is called attentional set shifting task, that actually made us realize that these guys, they're not really cognitive defective, but they have more of a problem of cognitive flexibility rather than comprehension of the test. So how can it be that all of these functions are connected? So how can we start from an altered mini-puberty and basically conclude with a reproductive insufficiency, but also mainly the question is, how do we have these cognitive and psychiatric problems, and what could be the role of nitric oxide on those? And actually, the answer would come if we checked a bit outside the hypothalamus, because the most important population of Enos neurons is actually found in the hippocampus, is the more well-characterized population. And what we saw that was very interesting to us is that the activation of these Enos hippocampal neurons is actually also controlled by estrogens, because when we overectomized our mice at P12, so in the beginning of the mini-puberty, we saw that the phosphorylation of the Enos neurons at P23, when normally the levels are supposed to be pretty high, as you can see here, was actually dropping, meaning that we were altering the pattern of the activation of the Enos neurons in the hippocampus through the exhaustion of the gonads. And this became even more interesting when we realized that there are many cells expressing the GnRH receptor around the region of the hippocampus and the cortex. And this was something that we did not know till very recently. And here you can appreciate in a 3D disco that basically the GnRH neurons that you can see here in white in a completely transparent mouse brain, they are sending their projections a bit all over the brain, including the hippocampus and the cortex, opening thus the possibility that players of the hypothalamic pituitary gonadal axis, they are not only found in the hypothalamus, but maybe they can communicate between each other also in extrahypothalamic areas. And when we use the paradigm of a mouse model of Down syndrome, so we went to a disease that has been linked with cognitive dysfunction and has never been linked with reproductive dysfunction, and we checked what is happening with a hypothalamic axis, something that no one had ever checked before. And we actually realized that these mice had an aberrant profile of LH pointing towards an impaired hypothalamic pituitary gonadal axis with a decreased, basically, extrahypothalamic expression of this GnRH peptide. We performed a preclinical study that I will skip because we also have a clinical study that was performed in 7 patients of Down syndrome that were showing before a complete altered LH profile. They were given a GnRH pulsatile therapy, meaning the native peptide was given to them in the physiological amount, and we actually saw that restoring basically the pulsatile release of the GnRH was able to restore monitor the cognitive performance, the visuospatial function, the executive function, the verbal comprehension, but also, as you can appreciate here, the functional connectivity profile in these patients. So this was a very tiny study, but a very, very promising one. And if we go back basically to check what could be the implication of nitric oxide in those effects, well, nitric oxide is actually able to control the electrical activity of the degenerate neurons, being able, basically, this is a nitric oxide donor, being able to inhibit the degenerate neuronal firing. And when checking what is happening during mini-puberty in the enos deficient mice, you can see that the absence of nitric oxide from the neuronal form is actually creating this increased spontaneous firing frequency. And when we checked what is happening in the transcription of the peptide, we saw once more the same type of pattern, meaning that the absence of nitric oxide was actually increasing by a lot the mRNA expression of the degenerates, meaning that everything corresponds to this exacerbated profile of mini-puberty with nitric oxide, neuronal form of nitric oxide, being able to control not only the electrical activity of the degenerate neurons, but also the transcription of the degenerates per se. So I'm going to conclude a bit here by responding to the question that maybe has been risen to you now. So if mini-puberty is so important, and if nitric oxide is so important, can we intervene? What would happen if we would restore mini-puberty in these enos deficient mice by basically restoring nitric oxide? We did that. So we gave either inhaled nitric oxide or sildenafil, which is the known Viagra, inhibiting the phosphodiesterase, so meaning that the cyclic GMP could be present for longer. In these enos deficient mice, specifically during the time window of mini-puberty, meaning from P10 till P23, and then we stopped. And we went in adulthood to check what is happening. First and foremost, we validated that the treatment was able to restore mini-puberty, meaning dropping these exacerbated FSH levels that we had seen in the enos knockout, meaning that we had come and we had normalized mini-puberty. What was the impact of that? Well, surprisingly, we saw that we managed to restore the delayed in the puberty in both females and males. And also we restored all the non-reproductive comorbidities that we had witnessed before, meaning that now the mice that had been treated only during the time of mini-puberty, that were still enos deficient, were actually performing the tasks as a wild-type animal. And this basically means that mini-puberty is essential for the proper functioning of the hypothalamic pituitary gonadal axis, supporting basically this progression from the infantile times to a more fertile phenotype during puberty, but that this axis and the maintenance of the degenerates production in a controlled, sustained physiological manner plays a crucial role for brain neurodevelopment and overall brain maturation. So it means that basically the role of mini-puberty will extend beyond the reproductive sphere, being key for the development of both mental but also non-mental functions. And this opens many, many possibilities for clinical trials, that some of them are ongoing. Why? Because we have cases of humans with this altered mini-puberty and comorbidities. So could nitric oxide be a possible therapeutic? And I'm mainly interested in drawing your attention to the preterm population of vulnerable kids that actually saw this same exacerbated profile of mini-puberty, and they show a high risk of different type of comorbidities as adults. So this population is being studied during in the context of this Mini-No-European project, where basically we assess the role of nitric oxide in the control of this mini-puberty and its possible use as a therapeutic. So I would like to thank many, many people who have collaborated for this study. Our team, here as you can see, my group is a small group in this big team. The human cohorts that we use for the preterm study that I could not present to you today because the project is ending next year, so maybe if everything goes well, next year I could present you this study. And obviously all of you for your attention. Thank you so much.